What can we do, eh? So today we're going to be talking about the immune system mostly, and a little bit about the lymphatics. The lymphatics are a series of vessels. And we haven't talked about blood vessels, but there's some similarities to lymphatic vessels to veins in terms of the way that they're organized. They have an inner layer that is very smooth to allow nice, easy passage, in this case for lymph fluid. And they have a middle layer that has a little bit of disorganized smooth muscle associated with it, which helps adjust the diameter of the lymphatic vessel, and then otherwise depends on some one-way valves and skeletal muscle contraction relaxation cycling to push lymph along its length. So generally, the lymph vessels look like these green vessels here. So you'll see that they're all over the body. Okay, and what their responsibility is to collect lymph, and lymph is going to come from capillaries. So I'll show you that in just one second, but just to get a little bit of an idea, these lymph vessels are going to be found all over the body. And if we followed them, we could follow lymph from, for example, like this knee joint area, and it would be traveling upward. And there it's going to travel through a node, and often will be connected to other nodes. Here's an example of some other nodes as they travel back up towards the heart. And ultimately, the lymph vessels will return lymph fluid back to the circulatory system. Here we look at these blue vessels are veins. So we're carrying the lymph and we're returning it from all over the body and emptying it back into these veins. These veins are just below the clavicle. So the name of the vein is actually called the subclavicular vein or subclavian vein on both sides, the right and the left one. So that completes the circuit. They're going to come from the peripheral tissues. So they might start right here, draining the finger area and traveling back towards the heart. So we're going to find these vessels all over the body, wherever there's oh, look, I drew these little arrows all over this screen. Let's get rid of those. So what we would see is if we had a blood vessel that carried oxygenated blood to a tissue, when it gets down close to the cells, it branches. And when it branches, it's going to become leaky. The capillaries now, and they branch out and they surround I'm just trying to give you some idea of the branches. And then what's going to happen is on the other side, you can't really see that very well. On the other side, we're going to have the veins. So we draw them in blue because they're deoxygenated. They delivered the oxygen to the cells. So the cells would be between these tissues. In this case, these. So what the circulatory system does is bring blood that's high in oxygen and high in nutrients to the cells. So we're heading in that direction. The blood flow is heading in that direction and being taken away in this direction by the veins. And we're going to have this blood will be relatively high in carbon dioxide and wastes. So the regular circulatory system is responsible for delivering oxygen and nutrients to all the cells in the body and removing carbon dioxide and waste from all the tissues in the body. We okay with that? Does that make sense so far?
Yes.
Okay, so one of the things that's going to happen is when we force nutrients and oxygen out of the blood, we also push water out of the blood. Blood volume is going to go down in the capillaries, and it doesn't all get picked up by the veins. So at every capillary exchange, we lose a little bit of water volume. And that means we're going to lose blood pressure. And when we talk about the cardiovascular system next semester, you'll find out that if we lose blood pressure, we're going to lose efficiency of delivery of these oxygen and nutrients to all the cells in the body. So we need to maintain blood pressure. And that's where the lymphatics come in. So we're going to have a lymphatic vessel that is down here at the capillary network, and it's going to pick up the extra volume of fluid that's lost at capillary exchange and deliver it back to the circulatory system. So we're going to go back, and in this case, we're going back to the subclavian veins. The veins all drain back towards the heart, and the subclavian veins are no exception. So what we've done is we've completed a homeostasis of blood volume with the addition of the pickup from the lymphatic vessels. So we say that when we look at the lymphatics from a functional point of view, one responsibility that they have is to restore blood volume by picking up lost fluid, or let's put it as plural, at capillary beds, and then returning it to the circulation. So that's a really important function of the lymphatic system. Now you can also see in this little drawing that we've made that the lymphatics are not an actual circulation. They start at the capillaries. They pick up fluid at the capillaries. And the way that they're designed to do that is that the end of the capillary, of the lymphatic capillary, is kind of a loose set of flaps. Before we run into the actual lymphatic vessel. So when the pressure, if there's higher pressure outside than inside, the fluid is able to run in these flaps. That's how the lymphatics will fill up. We okay with that?
Yes.
Okay. And then if we reverse that, as the flap fills up, then the pressure inside becomes greater than the pressure outside, relatively speaking, and that pushes all these flaps closed. So these flaps will be pushed shut, and the lymph that is in here is going to start to be pushed along the length of the valve. And as we move along at least the length of the vessel, and as we move along the length of the vessel, we've got these one-way flaps, these one-way valves that allow flow in this direction. But if the flow tries to go backwards, it shuts the valve down. So we get one-way flow that's heading back to the subclavian veins that are up near the heart. They're up in the thoracic cavity. So we're going to draw back towards the heart in this way, using the pickup at the capillary network and the one-way flow because of valves. And on either side of these lymphatic vessels, we've got muscles. And every time a muscle contracts, it gets fatter and it squeezes the vessel. And that's going to squeeze in a way that will push the lymph through the one-way valves and they can't come back. So we get, the more active we are, the more movement there is in lymph. That's called a skeletal muscle pump. You'll talk more about the skeletal muscle pumps when you do the circulatory system, because that's how blood moves in veins as well. We don't need a skeletal muscle pump for the arteries because the pump that drives blood through the arteries is the heart. So we've got one major function here that we've just drawn about here, and then another major function for the lymphatic vessels is that they transport some of the dietary lipids. Remember in portfolio number 1, you talked about some dietary lipids, lipids that are required for our diet. We have a couple of essential fatty acids. We can't synthesize essential fatty acids, so we need to get them from our diet. That's one of the reasons we eat fat. So when they're absorbed across the digestive system, which is a system we're going to start to talk about next week, they're one way that they're transported is by these lymphatics. They're going to pick up some of these larger fatty molecules that have been absorbed in the small intestine into the lymphatic vessels and returned to the circulation in the same way that the water was, the fluid was at capillaries, but starting in the digestive system. So we transport dietary lipids from the small intestine back to the subclavian veins. The same pathway. Once they're in the lymphatic vessels, they're going to follow the same pathway. So they're going to be introduced to the subclavian veins. And this is one way we get lipids, some of the lipids. We'll just say up here, it's some, not all, dietary lipids from the small intestine to the circulatory system, so that all the different cells in the body can use the fatty acids for the production of what sort of things do we produce from fatty acids in the body? Anybody remember? And in this case, it's something we talked about. We didn't talk long about it, but we did talk about it. It was on portfolio number 1. What do we use lipids for in the body? What do we resynthesize fatty acids into in the body?
ATP.
Triglyceride.
Triglycerides?
Yeah.
Yeah. So the triglyceride is partly carbohydrate. The glycerol part is a carbohydrate, and the tri refers to 3 fatty acids attached to the glycerol molecule. So that's a good example. And triglycerides can be broken down. I heard somebody mention that they are what we are going to be able to fairly easily break down into ATP. So that's very, very important as far as that goes. What else? What were some other examples? We talked about 3 main groups of lipid-based molecules. Triglycerides was one. Right, those were the big 3: phospholipids, cholesterol, and triglycerides. Cholesterol is an important building block, contributes to plasma membranes of every cell in the body, and also contributes to, for example, the production of some of our lipid-based, steroid-based hormones, usually ending in ONE, like progesterone and aldosterone and testosterone, all examples of cholesterol-based hormones. And phospholipids, where do our phospholipids play a role?
In the bilayer of the cell.
Yeah, for every single membrane, of every cell we produce is mostly made from phospholipids. So that's a pretty important dietary source, lipids, in order to resynthesize literally for every cell in the body. And there's trillions of those. So we need these dietary lipids for these purposes alone, as well as the other ones, but those are the big ones. Those are the big ones right there. And the third function for the lymphatic system is that it's got immune responsibilities, which is the topic that we want to focus on today. So immune responsibilities within the vessels and lymph organs associated with the lymphatics. Nodes and spleen are the big ones. But we also have something called MALT. So that's an acronym for mucosa associated lymph tissue. TLAM, MALT. So mucosa-associated lymph tissue is literally what it sounds like it is. This is mucosal tissue. Remember, mucosa was tissue that was going to be found lining entry points to the body, or if you want to think of it the other way, exit points from the body. But there's going to be a mucosa, double layer with a little bit of mucus associated with it. And these tissues are going to be high in lymphatic functions, immune functions. There's going to be a high concentration of white blood cells that we'll be talking about a little bit later today, and white blood cells that are in the recording on blood, because as we'll discover, the white blood cells are produced initially in the bone marrow and introduced to the circulation for distribution from there. Lymphatics and blood vessels are going to distribute these white blood cells all through the body. So these are the responsibilities of this lymphatic system. So we understand that it's a series of vessels that really move in one direction only. So that comes back to this picture, where we look at this picture here, and we appreciate that the movement that's occurring is going to be a collection. There's a lymph node, and traveling up through another lymph node and a chain of lymph nodes here. We're going to go through a series of lymph nodes. There's a whole bunch of lymph nodes before we empty back in to the circulation. And we see an arrangement here where we're draining from the peripheral areas, the areas far away from the heart, back towards the heart. Right? So whether we're talking the right arm or the left arm, we're going to go through series of nodes. These are nodes all in this area here. This is the armpit area, or known as the axillary area. These are the nodes that get involved in breast cancer. And we'll find out that cancerous cells are just like bacteria and viruses as far as the immune system is concerned. So it becomes what we call a non-self cell. And it doesn't matter whether the non-self cell originated outside of the body, like a bacterial cell would, or originated inside the body, like a virus-infected cell or a cancer cell would. They're going to ultimately, hopefully, be picked up by lymphatic fluids, and there's a number of ways that can happen, and travel through nodes where they're going to be exposed to white blood cells that are going to recognize them as dangerous and deal with them. And that's what we'll be talking about, what that means by dealing with them and that sort of thing. So that's the role from a lymphatic perspective. And we're going to drain back to the heart and ultimately, hopefully, deal with any of these viruses or bacteria or cancer cells before they get a chance to go into the circulatory system. If they end up, a virus or a bacteria ends up in the circulatory system, we're in a little bit of trouble, because now the circulatory system can spread those bacteria and viruses all over the body. and makes the immune system's job a lot more difficult than it is while it's in the lymphatic vessels themselves. Okay, so that gives us a little bit of an overview. Now, what we want to do is we want to look a little bit closer at this lymph node. So we've got a lymphatic vessel that's going to be picking up lymph at the tissues, and then it's going to drain the lymph with one-way valves all the way along its length until it enters a lymph node. Now we're going to draw the lymph node in the shape of a kidney bean. So the lymph node has got a convex surface that's bigger than the concave surface. And you'll notice that we have more vessels entering the lymph node than leaving. And within the lymph node, we're going to have clusters of white blood cells. And they're arranged in segments, but there's a high concentration of these white blood cells. These are our immune cells, so we want a high concentration of these immune cells here. And as the lymph is collected and travels to the lymph node, whatever is in that lymph, whether it's a cancer cell, whether it's a virus or a bacteria, it's going to be exposed to all these white blood cells when it gets to the lymph node. And in our little drawing here, it could be 5 vessels entering, but these vessels that are carrying lymph towards the lymph node are called afferent. We should be okay with that terminology. We know that in the nervous system, the afferent neurons carry towards the central nervous system. In this case, the afferent lymph vessels carry towards the lymph node. And that makes this vessel called a what?
Efferent.
Efferent.
This one will be an efferent vessel. And we can see that if we have 3 or 4 or 5 afferent lymphatic vessels entering a lymph node and only 1 or 2 efferent lymphatic vessels leaving, what's going to happen to the lymph once it hits the lymph node? Is it going to be slowed down or is it going to be sped up? If we have lymph volume traveling into the lymphatic node in 3 vessels, the lymph is going to be spread around.
Slow, right?
What's that?
It will slow because it has to stop briefly before it shoots through, like it'll bottleneck.
Exactly. It's the way to describe it. So with a bottleneck, it's going to slow down. And what is that going to do? That's going to give us more time for exposure to the white blood cells, more time to recognize a non-self cell, right? In the immune system, we talk about self and non-self all the time. So self is our own cells with our own identifying markers on them. And non-self is anything else, whether it's a piece of wood, whether it's a piece of plastic, whether it's a virus, a bacteria, or a cancer cell. Those will all have different markers on them to identify them as non-self. So these white blood cells are on the lookout for non-self, but they're not very specific. A lot of them are not very specific. So we're going to be dependent on some time for exposure. So one of the ways we build time is by having more afferents than efferents. So it's going to slow down lymph. And the other advantage, or the other thing that we see, and we saw that in that first picture I showed you, is that in a lot of cases, these lymphatic vessels are coming from another lymph node. So this lymph node has got a huge millions and millions and millions of different white blood cells in here. All kinds of different white blood cells, names that we'll be talking about. There's B lymphocytes, and there's T lymphocytes, and there's helper T cells, and there's neutrophils and eosinophils. Some of these names, if you've looked at the recording on blood, that we named those cells in that recording. But for now, we're going to be probably learning some of those names for the very first time. But you can see that what's going to happen is these are now afferents, and this one represents an efferent from this lymph node, but becomes an afferent for this lymph node. So if some of the lymph that's in here escapes with some virus cells, let's make the virus cell this orange color. So, well, a virus is not actually a cell, but let's say we're carrying a virus into the lymph node. If it somehow got past this lymph node, it's going to be delivered to the next lymph node. And it's probably not going to get through all the lymph nodes in a chain. So these lymph nodes are typically linked in a chain. So we call it a chain because they're going to be linked together. So there's, for example, in the armpit area, there's an axillary chain. In the groin area, it's called an inguinal chain. There's a chain in the neck, cervical chain. There's a chain on either side of the aorta in the abdomen area, abdominal chain. So these are all places where we would see a linking of lymph nodes with the goal of creating a bottleneck. Perfect name, great name. I haven't used that name yet, but I usually just talk about the lymph sort of being jammed up because we've got more afferent vessels going into a lymph node than leaving. But bottleneck is a perfect way to describe it. So we're bottlenecking in each of these lymph nodes. That's increasing the chances of exposure. And if somehow we escaped on the first node, we're probably going to get caught on the next one. So when we look at this picture, we've got these chains of lymph nodes. There's a lymph node, there's a lymph node, that's axillary on either side of the aorta. So the aorta would be traveling down right through them, right as it leaves the heart. The aorta is going to come like this and travel right down the center. We've got a chain on either side there. And there's a chain in the groin area on both sides. Those would be the inguinal ones. And we've got cervical ones. The cervical ones are the ones you've probably felt before. If you've ever had a good cold, maybe some of you got coronavirus during this crazy pandemic of ours. But when the lymph nodes are active, because they've been exposed to a virus or a bacteria, or a cancer cell for that matter, they are going to increase in numbers and multiply and cause the node to swell. And the node will get larger and often tender. And when it's up in the neck, you can feel them. You can feel that you have swollen nodes. And, you know, sometimes it's like a lot of things in healthcare. Initially you think, oh no, I'm in real trouble. But in reality, what it's telling you is your lymph system is doing its job. It's multiplying in response to the exposure to some sort of non-self cell or group of you know, we call them antigens. I'll write that word down in a minute. But this is the organization of the lymphatic vessels. So when we talk about lymph tissues, we can talk about primary lymph tissues and secondary lymph tissues. So the other thing to realize is that with lymph nodes, we have, you know, let's see, what slide am I supposed to be on here? Somewhere in the vicinity of 500 to maybe as many as 900 nodes in the body arranged in these chains. And we'll have a few individual nodes that aren't really obviously in a chain, but ultimately they're going to drain into the chain. So these nodes are going to be funneling in the lymph, creating a bottleneck for more exposure. And this is the location where a lot of our immune cells are going to be found. So we have a chance to recognize the non-self cells and act against them in these lymph nodes. And a whole group of lymph nodes, or sorry, a whole group of cells in the lymph nodes are phagocytes. And we know that phagocytes are what? What's a phagocyte do?
They eat.
Yeah, they eat. So if they're big, we'll call them a macrophage, a big eater. And if they're other groups of white blood cells, like a neutrophil, is a phagocyte. It's an eater. So ultimately what we want to do is eat this virus or this bacteria, because once we get them inside that phagocyte, we can act on what's gone inside that phagocyte with enzymes and destroy it, and either break down the virus or the bacteria to amino acids and monosaccharides and fatty acids and rebuild with those, or eliminate them from the body altogether. So that's the ultimate goal of any of these attackers, any of these non-self cells. So we have primary lymph tissue. And primary lymph tissue refers to the tissues that the lymphatic, the lymph cells, white blood cells, originate from and mature from. So the most common primary lymph tissue is red bone marrow. All our blood cells are produced in red bone marrow. It doesn't matter whether you're talking about red blood cells, white blood cells, or platelets. All of the cells associated with what circulates in the blood are going to be found originating in red bone marrow. And again, the recording that you've got on blood explains this as well. So our white blood cells are no exception. They're going to originate here. But also, there's an organ called the thymus. And the thymus is the other example of a primary lymph tissue. The thymus is responsible for maturing T lymphocytes. So the T lymphocyte is produced in the red bone marrow, but it's going to be matured in the thymus. So T for thymus. So there's another class of lymphocytes that are called B lymphocytes. They're going to originate in the red bone marrow and mature in the red bone marrow. But T lymphocytes have to be transported to the thymus for maturation. We're going to talk a little bit later about what that means, what maturation means. But basically, pretty simply put, it means learning to recognize the difference between self and non-self. And we'll talk in a bit more detail about that. So that's primary lymphatic tissue. If we're talking about primary lymphatic tissue, we must also be going to consider what? If we differentiate, yeah, well, there must be something that we're going to refer to as secondary lymph tissues. And the good news is we've already talked about one of those. Those are the nodes. So they're secondary because the cells have been developed in the primary tissues, and the secondary tissues are the location for many of these cells in the body. So we know that at the lymph nodes, we're going to see clusters of white blood cells and B and T lymphocytes in these lymph nodes waiting for some exposure, delivery from lymph for exposure. But the tonsils The tonsils really are an example of the mucosa associated lymph tissue. There's an area in the small intestine called, unfortunately, it's still got a guy's name associated with it. And that's also a mucosa associated. It's called Peyer's patches. That's a cluster of white blood cells we see in the small intestine. And the spleen is another example of a secondary. It's the largest of the lymph organs, and it's got a large concentration of white blood cells there, and it's responsible for lots of functions associated with the immune system. A lot of lymph is going to flow through the spleen. It's a very vascular organ, so exposure to blood and the white blood cells that are being collected in the blood and delivered to the spleen too. So the spleen plays a very important immune function as far as that goes. These are describing secondary lymph tissues. Primaries is where the cells originate, and secondaries is essentially where they're going to collect and act when they've recognized a non-cell cell. Okay, so we've got this sort of leaky cap that collects blood volume and collects, and the volume of blood that's collected is then referred to as being lymph. When it's inside a lymphatic vessel, it's called lymph, and the destination is through lymph nodes for exposure to white blood cells, and then ultimately returning the volume to the circulatory system for those functions that we talked about at the beginning, the three main functions of the lymphatic system. Now, when we jump to the immune system and the functions of the immune system, there's a basic principle that we want to be aware of. I've mentioned it, so it's not going to be a surprise. When we talk about immunity, it really isn't a nice clean system. You know, when we talked about the skeletal system, it was a series of bones that link together to create joints, bones that allowed movement to occur. The muscular system was pretty straightforward. You know, there was groups of muscles that had functions that they were responsible for, and that sort of thing. Later on, when you talk about the respiratory system, it's about the lungs and the airway. The cardiovascular system is about the heart and the blood vessels. But the immune system is kind of messy. The immune system is a whole bunch of lots of different things. So we've got processes. And when I say processes, I mean things like inflammation, and fever. We've got cells. Most of those are white blood cells with names like neutrophils, dendritic cells, T and B lymphocytes, etc. Cells. And we've got chemicals. Proteins with names like interferon. What a great name for something that's going to block or interfere with something. It's simply called an interferon. We've got some other proteins called complement. We've got some proteins that are known as antibodies. Okay, and all of these things work together to hopefully recognize and activate against non-self cells. So when we talk about the immune system, it's a big, big system. There are literally trillions of cells that will be involved in these processes. The processes of inflammation, of cell activation, chemicals that are going to play a role in messaging, as well as action against non-self entities, whether they're cells or fragments of cells and so on. But it's complex. It's complex. And when we talk about this immune system, so one thing to keep in mind is it's a big and messy system. And this is a quote from the people, if you've had a chance, I set up a little quiz on the immune system. I don't know if you've had a chance to look at it yet, but there's a video, and it's sort of a colorful video, and it's a description of the way parts of the immune system work. But that guy wrote a book about the immune system, and that's how he sort of describes the immune system. And it's a good way to think of it. It's big and messy because it has all these other things. But the one thing that's in common is that our goal is to be able to recognize and then activate against a challenge. And that challenge is typically going to be from a non-self cell. A non-self cell is by definition a threat to the self cells, because by definition, non-self cells want to mess up the self cell's responsibilities, either by taking over by mass size. That's what happens in some cancers. You know, some of your cancers are described as benign cancers. Benign cancers are just cancers where cells grow in mass and they're not spreading, but they're going to cause problems because they get big. You know, you'll hear about somebody who had a tumor as big as a grapefruit taken out of their abdomen. Well, if you've got a mass growing in your abdomen that's as big as a grapefruit, it's got to be pressing on other organs and tissues in the body. And if it presses on a say, for example, a kidney, you may lose kidney function, right? So the cancer itself is not causing a problem except by its size. You follow me on that?
Yes.
So our immune system does a really good job at dealing with cancer cells normally. On a daily basis, there are abnormal cells that have undergone mitosis and mutated and are potentially cancerous. But our immune system, through the lymphatic vessels, is really good at picking those up and dealing with them, getting rid of them early. A lot of cells, once they mutate, they will undergo what's called apoptosis, which is a fancy term for cell suicide. It's a nice, clean way to get rid of abnormal cells, happening all the time, daily basis, to the tune of millions of cells on a regular basis. So when we think about our immune system, really, we want to keep our immune system optimum, you know, by feeding it with good nutrition and exercising on a regular basis and having good sleep habits. All these things maximize the efficiency of the immune system. We can already see that surveillance is important. So we're going to talk about some cells that have a primary responsibility of looking out. And then we want to also understand what we mean by that term activation. So it's a big and messy system. And the other way to think about it is that it's an interlocking multi-layered activation of one, two, and three. So we're not going to activate one cell and say, okay, you do your job. If you can't do your job, we're going to recruit somebody else. We are going to recruit, but we're not going to wait until the first group of actions has gone through a whole series. We're really literally going to throw the kitchen sink of the immune system at the entire problem. So we're going to see really what we would describe as positive feedback in an immune response. when we get recognition, we're going to recruit immediately every tool in the immune system we have available to us to activate and destroy the possible threat from those non-self cells. So it's interlocking, multi-layered activation. It's not first one and then the other. So we divide the immune system generally into what's described as innate and adaptive, or specific and non-specific. But it's easy for us to imagine that maybe the one goes before the other. But the reality is that's not the case. We're going to activate and recruit all tools we have available to us to get rid of this non-self threat. So it starts with a definition. An antigen, that we're going to abbreviate as AG, is any potential threat to the body, anti-genesis, anti-growth and development of the body. That's really what the antigen term refers to. And an antigen, to be an antigen, is going to have two primary properties. One of those is called immunogenicity. So this is really non-self. Okay, so when we talk about immunogenicity, this is a property that allows the immune system to recognize the foreign invader. So this immunogenicity is all about recognition. And the second property that we expect to see in a typical antigen is the property of reactivity. And reactivity is the ability of the immune system after recognition to activate against it. Activation. And another important, I guess, verb in this case is the term proliferation. And we'll talk a lot about proliferation when we get to the adaptive type of immunity. But really what we're talking about here is making more cells that will recognize that specific antigen. So that's going to help us for future exposures. So one of the things that we hope to have happen in a vaccination, thinking about coronavirus, is that we're going to make specific new cells that will recognize coronavirus. So the first exposure is dealt with through steps that involve inactivation and proliferation. And a second time that we're exposed, we've got now a whole army of specific cells that will recognize coronavirus the next time it's exposed to us and deal with it very, very quickly. So that becomes an important step in the immune response. So the antigen is against us, and it's got to have properties that allow us to have recognition. And usually we can appreciate from what we've talked about that there's going to be cell markers, protein sequences on the surface of a cell that differentiate between self and non-self. So if we had a cell membrane, there's our lipid bilayer of a self cell, we would have markers on this self cell. Sometimes there's thousands of markers, but they're proteins that are found in a sequence. So here we have a series of proteins. We've got purple, yellow, green, and this sort of orangey red color. So we got a series of markers in a sequence, and I'm just saying that in this example, that represents a self cell. Any other cell in the body that has any other sequence. So the sequence is that. So we've got this sort of fuchsia color, I guess. Then we've got our orange. And then we've got this turquoisey color. Now we've got a cell, a white blood cell that is trained or is developed in a way that when it sees this cell, it does not activate. It does not recognize that cell as a danger. But when it bumps into this cell, it's got receptors on it. Now, if it's a specific immune cell, we'll get to that in a minute, it might have a set of receptors that's designed to match up with that specific cell and activate against it. So that's what we're talking about when we talk about recognition. So we have some cells in the body, some immune cells in the body that have pattern recognition receptors. So these are non-specific. They're basically capable of recognizing a self pattern and leaving it alone, and then being activated against anything that's not self. That's pattern recognition. We have other immune cells that are specific. In this little drawing I've given you here, that's an example of where we would have a specific cell for a specific virus or a specific bacteria or whatever. And we'll see when we get to the adaptive immune system, it's really kind of a miracle in a lot of ways, because we've got a cell, a white blood cell, whether it's a T cell or a B cell, for every possible protein sequence in the universe. And we produce those first in the red bone marrow and mature the T lymphocytes in the thymus for every possible virus or bacteria known and not yet known in the universe based on proteins. We've got a limited number of amino acids, about 20. And we can sequence those 20 into hundreds of thousands of different combinations to create proteins and then put those proteins in different combinations. But we've got a cell for every possible match. There's a cell for the Ebola virus. There's a white blood cell for coronavirus. The problem is on a first exposure, these steps up here take some time. So on a first exposure, you might die. But for most first exposures, you get sick. And the sick that you're going to get is based on the reactions of the immune system to the tissues that are being attacked. So inflammation and fever and actions from cells are going to present some symptoms. And that's what we want to talk a little bit about when we look at how the immune system responds. Are you with me okay with this so far?
Yeah, that makes sense.
Okay, so the thing to realize is that on a daily basis, on a minute-by-minute basis, we're going to be exposed to hundreds of millions of bacteria and viruses and other non-self organisms like, like parasites and worms and even, cells that come from plants that might irritate. Like a sliver does when you get a piece of wood stuck in your skin and it creates an injury that results in some inflammation. These are all types of challenges that the immune system needs to respond to. And cancer cells, abnormal self cells that mutate and become a threat. So these are all things that become important for the immune system to be able to recognize and deal with. So we divide the immune system loosely into what's called innate, and sometimes we say that's non-specific immunity, and then we have adaptive, which is going to be specific immunity. So we're going to talk about innate immunity. And when we talk about innate immunity refers to the type of immunity that you're kind of born with. Okay, so in innate immunity, these are any and all defenses. So all defenses that we are born with. So some of our white blood cells are ready to be active right away. Neutrophils and eosinophils, basophils, mast cells, these are all examples of white blood cells that are going to be part of our non-specific response. We're going to talk a little bit about some of those in just a few minutes. So we're born with these, and they can be activated within seconds of an exposure. So they're very, very fast, but they're very general, which is non-specific. So they're going to operate the same way. So think about inflammation, okay? Inflammation will operate the same way whether the injury was caused by an infection or by mechanical injury, like that sliver. The inflammatory response will be the same regardless. That's a type of non-specific defense. When we look at adaptive immunity, we've got specialized cells. And that's those T lymphocytes and those B lymphocytes that are specialized and specific. So they have receptors for specific antigens. Whereas in our non-adaptive, we've got recognition, pattern recognition receptors that are basically set up to recognize the difference between self and non-self. So it's very general. It's very important, but it's very general. Whereas in the adaptive or specific version of the immune system, we've got cells that are specific. So if you think about flu virus, some of you may be aware that, flu viruses have subcategories. We sometimes categorize them with numbers and letters, H1N1, H2N5. So the idea is we've got a cell that will recognize H1N1, and we've got a different group of cells that are designed to recognize H2N5. A different group of cells for every possible variant. And the same concept applies to the coronavirus, the family of coronaviruses versus the family of rhinoviruses. There's different groups out there. Bacteria, same idea. But we do have a cell that will match and activate against any protein sequence that's non-self. And in the adaptive immunity, the beauty of being specific and it's also described as being systemic. And the beauty of adaptive, the key thing here is that we get some memory. We develop memory cells for the specific antigen that it was matched up with. So if we go back up to this picture and we say that this is a virus infected cell or a bacteria, and this one over here was self, what's going to happen in the adaptive immune system is that we're going, at the end of activation and proliferation, we're going to have memory. So we're going to have new cells, a larger number now of cells that will be ready for a second or a third or a fourth exposure. And we'll reinforce that when we look at the specific immune system in a little bit more detail. So let's look at some innate mechanisms. Innate immunity. So when we talk about innate immunity, in some older textbooks, they talk about first, second, and third lines of defense. More recently, we talk about them just in 2 lines of defense, but we'll keep them as 3 because, well, we'll just not talk very much about first and second being separate than third. We'll just talk about the mechanisms that are included under innate immunity. So remember, these are non-specific responses. So they're going to fall into a couple of main categories. When we look at innate immunity, we're thinking barriers. And then we're thinking about some cells and processes and some chemicals that respond in a non-specific way. So when we think about barriers, what's the most obvious barrier that we have available to us as a type of non-specific defense?
Skin.
Skin.
Yeah, the skin. So think about what we learned about the skin, the epidermis. Now everybody's going, oh no, we're going to talk about the skin again? The outer most layers, and there was anywhere from 30 to 50 dead cells. Layers, 30 to 50 layers of dead cells. So what better barrier can you have than a whole bunch of dead cells? Viruses need live cells. So a virus that lands on your skin has got a huge disadvantage right away. Not mostly because it needs live cells to survive, and it's not going to be able to do that. So when a virus gets inside the lungs, like coronavirus does, it's got access to live cells, but when it lands on your skin, not so much. Remember way back early in the protocols for the pandemic, one of the things they said was, wash your hands constantly, don't touch anybody, go elbow to elbow, all this sort of thing. Well, the reality is, and we found out later, that's not a necessary protocol for the coronavirus, because the coronavirus was airborne and you breathed it in to get into trouble. It wasn't going to effectively survive on your skin for any length of time. So that protocol kind of got dropped a little bit later, right? The masking and the distancing, they made sense for an airborne antigen, in this case, the coronavirus. But also, the epidermis was a type of epithelial tissue, was avascular. No blood supply, no mode of transportation for a virus or a bacteria when it sits on our skin. There's no way for it to travel deeper into the body if there's no blood vessel that it can access. And what else do we know about the epidermis that's going to provide a defense? Sweat and sebum. Remember, sebum was the oil produced by sebaceous glands. That tends to be slightly acidic and has enzymes that are antimicrobial. Antimicrobe is a general term for it, anything that's microscopic, and in this case is non-self. So these are all examples of general or non-specific immune mechanisms. This is very passive. This is about the thickness of the skin, the layers, the avascularity of the outermost layer, and the composition of sweat and oil that's sitting on the skin. All of these things make exposure less favorable. Now, at the same time, most of us recognize that we have a large colony of mostly bacteria that grows on our skin naturally. And that colonization of bacteria that grows on our skin naturally is our microderm, or our microbiome, is beneficial to us most of the time. But it's beneficial to us most of the time when it stays on our skin, not when it gets inside the circulation. So when we get a cut or a wound, it becomes potentially a problem, because the bacteria that was growing on our skin, minding its own business and really helping us, is now going to potentially be exposed to the circulation and could cause us problems if exposed to the circulation. So these barriers are very effective most of the time. And if we break the barrier, we get ourselves into a little bit of trouble. So that would be one of our examples of a first line of defense, right? Barriers and mucous membranes. The mucous membranes produce the enzymes and the antimicrobial components that help make sure that entry into the body is limited, right, to this exposure to hundreds of millions of different bacteria and viruses every day. So what's an example of a process? Well, the best example, the one we'll spend a couple of minutes talking about, is inflammation. So we know that generally inflammation is going to involve some swelling and some heat probably and things like that. So inflammation looks like this. It's got swelling, it's got heat, it's got redness, and it's got pain. And sometimes you'll see they'll put in loss of function. But 1, 2, 3, and 4 are all pretty consistent with all forms of inflammation. We're going to get swelling, we're going to get heat, we're going to get redness, and we're going to get pain. So that's what it looks like. What's its purpose? The purpose is to prevent spread, so maybe we'll put the letters in here so that, and to clean up the source of the inflammation, whether that's injury or infection. We're going to activate some mechanisms that are going to clean things up. So that's cleanup. And the third advantage is that we're going to prepare, so this is otherwise known as localizing. And the third thing is we're going to prepare for repair. A nice little poetic line there, prepare for repair. So we like inflammation for all these reasons. So what's going to signal inflammation? What's going to start the inflammatory response? It's basically, in old-fashioned terminology, a chemical alarm. Chemicals released from damaged cells and later activated white blood cells. So the chemical alarm will be released from damaged cells. So the cell membranes will release chemicals. Some of these chemicals you've heard of, they're not important for us today or for test 4 for that matter, but they have names like prostaglandins and serotonin and histamine. Some of these you've heard of before, but these chemicals are going to initiate the inflammatory response. They are responsible for the chemical alarm. And then activated cells that are attracted to the site of the inflammation will release their own set of chemicals to maintain the inflammatory response until cleanup and repair has been completed. So that's where these chemicals are coming from. And these chemicals basically are going to cause vasodilation and increased permeability of the local blood vessels. So vasodilation, we're going to make the blood vessels wider and we're going to make them leaky. And you can imagine that when we make the blood vessels bigger they're going to come closer to the surface of the skin. That's where we would get redness. We can see the contents of the blood vessels under vasodilation. That's what happens when you blush, vasodilation. But we also increase the permeability of the local blood vessels. So at the site of the injury, we're going to see swelling, and blood is warmer than skin, so we're going to get heat. Now, some of the chemicals are responsible for pain. Chemicals like the prostaglandins and serotonin, they initiate a pain response. And the advantage of pain is that it brings to your consciousness the injury, right? So this inflammation is really, really actually a big first indicator that we've got a problem. And sometimes, especially in healthcare, we tend to think of inflammation as being a negative. Oh, we've got to use anti-inflammatories. We've got to get the inflammation under control. Well, acute inflammation is a big advantage for all these reasons that we're talking about right here. We like inflammation for all these reasons right here. Chronic inflammation is a different thing. Chronic inflammation is telling us that we didn't go to completion. We didn't get to repair. And that's a problem. That's something that we want to address. But inflammation in an acute situation is going to take care of the spread. It's going to initiate cleanup, and it's going to prepare for good recovery, for good repair. So we like inflammation for all those reasons. Now, inflammation also attracts white blood cells to the site of injury. Now, that injury, remember, it's not necessarily trauma. It could be infection, right? But in each case, the cell membranes have been damaged. That's what initiated the response in the first place. So neutrophils, they are one type of white blood cell that are going to be activated early in the response. So they're sometimes, and this is a very important point, they're nicknamed first responders. But if you're asked for a function to describe a purpose or a function of neutrophils, first responders is not an answer, right? First responders is just a little like a mnemonic, a way to remember that these are going to be the first ones to arrive at the site of the injury and begin cleanup, because they're the most numerous. White blood cells in normal circulation. So because they're the most numerous, they're going to be most readily available, and they arrive in numbers to the injury site first. Mostly because they're already there, they're in the circulation. And when we vasodilate at the site of the injury, we're going to draw more flow to that site of injury. Now, what do they do when they get there? That's the answer when you're looking at functions. So these guys are aggressive phagocytes. So they're the ones that are going to arrive first, and they're going to start the cleanup. But they don't last very long. They last, maybe a few minutes to a few hours before they die off. But one of the other things that they do is they release granulocytes. They're a cell that they kind of look, they've got kind of a shape like that. There's a nucleus, and they're filled with granules. You'll see in the video on blood cells that they're called a granulocyte. They're a cell that have granules. Well, when those granules get released, those granules are going to be able to degrade or break down what was eaten, degrade the phagosome. So the phagosome was what was ingested by phagocytosis. Let's just leave it at that. And one of the things that's relatively new that we understand that these neutrophils do is that they create what's been acronymed as a NET. It's a neutrophil extracellular net. Neutrophil extracellular net. It literally explodes the DNA and proteins inside, explode, and the proteins are enzymes mostly that are able to block and prevent spread. To trap, where did my, to trap and again degrade anything that's non-self in the area. So the source of the problem, virus-infected cells, bacteria that got in. If it was an injury that was caused from an open wound, then there's going to be bacteria brought into that wound by either the surface of the skin or the content of the soil, for example, if you cut yourself on the ground, that sort of thing. So, and the other thing just to be aware of generally, which isn't a major thing, but they act in groups. So they're sort of attracted to each other. And it's kind of like a swarm. So they will, once one group of neutrophils are activated, they attract other neutrophils and they all work in an area en masse. And it's a little bit like watching a bunch of bees, leave a hive and, go after a potential threat in that scenario. So that's one of the cells that's going to get activated during the inflammatory response. Later, so this is for minutes to hours. Later, monocytes arrive. They cross out of the blood vessel and become big eaters. And they're going to last from hours to days to weeks. They're going to finish the cleanup. So here's some examples of some non-specific cells, part of the non-specific immune system, that are activated during the inflammatory response to promote this cleanup. Okay, we're going to take a break.